This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 861, recorded on the 1st of February, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 44 degrees Fahrenheit, which is pretty warm Celsius also. I'm getting that up. 7 Celsius. But the big news is the big storm that's coming, it's going to start raining this afternoon and then turn to snow up to 14 inches by Thursday night. Yeah.
Wow. That's a lot of snow.
Probably, they're saying 8 to 14 inches, and 14 is probably over closer to Lake Michigan, where they get lake effect snow.
Wow. Well, we had about 6 inches on Saturday. Not bad, but it's very cold, so it's not thawing. It's just remaining. Mm-hmm. Although here in New York, of course, it's turned to brown mush, which you have to navigate when crossing streets. It's very slippery. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We're looking at 63 degrees and socked in. Totally cloudy. No Tinkerbell today. At least not for a while. I don't think ever. We're headed for 66. It's going to cool off tonight. Rain all day tomorrow. And then the temperature is going to crash with tomorrow night down to 21 degrees. And forecast for freezing rain and all sorts of awful stuff on Thursday. And my daughter Susan lives in Boston. And she sent some pretty awesome pictures of clearing out in excess of 2 feet of snow around her house.
That's from the weekend?
Yep.
Wow.
They got clobbered.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. We missed out on the big snow, which was fine with me this weekend. And now it's 37 Fahrenheit, 3 Celsius, and very sunny. I saw some similar pictures from my sister. That was quite the storm in Boston.
We have a guest today coming from This Week in Evolution, TWiV, from Salt Lake City, Nels. Welcome.
Hey, thank you. Great to be here. Greetings from LD Lab Studio. I've got a few weather-related comments to make out of jealousy, mostly. So Salt Lake wants to reclaim that 2 feet of snow that Rich was just talking about. Around these parts, we usually, in the winter, we would call that a Tuesday up in the mountains. And unfortunately, the high trough areas have been pushing all of the snow over the top onto the East Coast. So Kathy, I guess we're sending about a foot or so your way. Mm-hmm. Also would like to reclaim that as soon as possible. It's been dry here. So the temperature is 28, which I think on the centigrade scale puts us at, what is that, -1 or -2? And dry, sunny and dry. Looking for those storms to push through to replenish the mountains.
Wait, it's not 28. What did you say? -1, -2. Okay, yeah.
Yep.
28 is 82. Which is a different season.
28 Fahrenheit. Yeah, absolutely.
We have some cool science to talk about today, but first, a few announcements. As I mentioned last time, Columbia University, the Aaron Diamond AIDS Research Center is looking for an assistant manager for their biosafety level 3 facility. This is actually it's run as a core facility. In other words, people get trained to use it, and then they can use it. So I will put a link to that. You need to have some kind of experience and so forth, minimum qualifications, and perhaps someone in TWiV listener land would like to do that. The other interesting thing, for a better word, not a thing, it's an announcement, let's say. Last time I bemoaned the fact that I couldn't do a Amazon Associates, but it turns out there is this Amazon Smile program, which I didn't even know about. And Cody wrote in after the last episode, say, hey, you can register. And in fact, Microbe TV is already there. Someone put it in for you. So if you want to buy things at Amazon, you you go to the Smile place. We'll put a link, and you can choose your charity. I said to my wife, you could do this. She said, I give it to, what's the pet rescue thing?
The ASPCA?
Yeah, maybe it's something like that. She said, I'm not giving it to you. I'm giving it to them. I was, okay, that's a good cause. But you can pick your charity. So you can search for Microbe TV. And then we get like 0.5% of the value of your purchases. And it doesn't cost you anything else. And it comes to us. So the cool thing is I have a little dashboard where I can see how much, for the first time, I can see how much is coming over to the company. So right now it's 0. But if you would like to there are other ways you can support us. Microbe.tv/contribute. This is now embedded on that page. So thank you, Cody, for pointing that out. I really appreciate it.
And you'll put that on that general page that you have, support our work at Patreon, PayPal, and more. You'll have the link there as well.
It will be there, yes.
Yeah.
There's
Oh, yeah. It says when you shop at smileamazon.com. Yeah. Amazon. Okay.
Yep.
Cool.
I think you go there and then you pick the charity and then you shop, right, as normal. So
yeah. So basically, as long as you always go to smile.amazon.com instead of Amazon, you always are donating a percentage of your purchases. I also have had it for years, donating to a different charity. So maybe I'll switch it.
Right. And when you click on the link, Vincent, that you put on the Microbe TV page, and it now is coming to me, do I want to change my charity to Microbe TV from the current charity I'm donating to? So,
okay,
cool.
All right, cool. I was totally unaware of that, so that's why I sometimes mention things here, because there's someone out there for sure that knows it, and so that's that. All right, Kathy, what do you have for us?
Well, the moment we've all been waiting for, the ASV 2022 registration page is going live. So by the time this episode drops, it will be live. There's nothing holding it back right now except for some IT issues. But anyway, so you go to asv.org/ASV2022. There are a bunch of different links there. You can click on the registration link. So you can just go to asv.org/ASV2022/registration. That will also get you there. But
yeah, it's working.
I think you can figure it all out. So, but there'll be a big red button that says, instead of saying registration will go live February, which is what we're seeing today, it should say registration is, click here. So,
Kathy, and ASV is at Madison this year?
It is indeed.
Yeah, one of my favorite venues. That's where I met someone called Kathy Spindler for the
first
time.
I was just going to say, it's where I met you. In fact, I remember exactly where I was standing when I met you, too. So, yes, indeed.
I remember you were fishing at somebody's house on the lake, Nels.
Yeah, Rob Kalita had us over.
Rob Kalita,
I remember that party.
Yeah, that was pretty fun. This is like science living, where you are like literally living on the lake, fishing in the morning, maybe boat into work, and then do some research and sail home or something. Not too shabby.
In the winter, you could drive your car over it, right?
That's right.
Skate. Yes, indeed. All right. Onwards. So the reason we brought Nels here, we love to have all the other hosts are always welcome to go on in one of the other podcasts. That's the cool thing about having a nice bunch of scientists. We have a paper, which I think is Nels is the guru of molecular evolution. And so this paper is all about that. It's published in the Journal of Genetics and Genomics just this past December. It is entitled Evidence for a Mouse Origin of the SARS-CoV-2 Omicron Variant. Comes out of Beijing, two places in Beijing, the Institute of Genetics and Developmental Biology and the University of Chinese Academy of Sciences. Wei, Shan, Wang, Zhang, Huan, and Shan. And so we're going to go through this with Nels's incisive commentary to guide us clearly, which I could not do that well for sure. So thank you, Nels, for coming.
Oh, of course. No, it's a fun honor to be among some pure podcasters on TWiV. And I think this hits on a major question in all of, our pandemic living, where we're seeing evolution in real time. I do have a little bit of an ulterior motive, which is to hype a few things that Vincent and I have been doing on TWiV. We'll talk about that later, but let's dive in on this paper. And I think, to set the table here, with the Omicron variant in particular, if we rewind the tape to before that hit, I guess this is November, December. It only feels like 2 decades ago now in pandemic time, in the pandemic time frames. If you talk to most evolutionary virologists, they would have predicted that the next variant would emerge from within the diversity of the Delta variant, which was obviously massively successful through that wave. But then, of course, as we all know, there's sort of a curveball here, evolutionary curveball, which was Omicron coming out of the weeds, or maybe to put it a better way, on a completely different branch among the variation of SARS-2 viruses that were being sampled. And of course, this is all very recent history, discussed widely, first spotted in South Africa, which emerging from a kind of mysterious location, both genetically and the source of that as well. And so I think that's where this paper picks up, is to ask kind of a provocative question, which is could it be possible that the Omicron variant actually emerged from mice? So the set of events here would be, obviously we had the 2019 spillover, probably from the wet markets. And then this thing circulating in humans for a while, sort of quietly under the radar. There's a reverse zoonosis event from humans to mice, or another rodent, or some source like this, and that's what we'll get into a little bit here in a minute. And then it comes back, and that then marks the emergence of Omicron, that sort of surprise variant that has swept the globe in remarkably short time. And so, I think 3 hypotheses that have been put out there. One is this one from a mouse, and we'll look at some of the evidence, the sequence comparisons they do, to think about some of the evidence there. The second is, and I added a short kind of case study that just came out a few days ago from South Africa, that might kind of put the brakes a little bit on this somewhat complicated reverse zoonosis and then re-zoonosis event. And then I think there's a third hypothesis. Actually, does anyone, what's the other possibility that I'm forgetting?
I think, so my understanding was that the reason why this was so interesting was that there's this very long branch where Omicron is sort of not directly related to other previous circulating viruses. And so I guess at least one other piece of this is just that there is some other place that there is circulation where we're not sampling. And so perhaps it's a question of lack of sequencing in some parts of the world where all of the precursor viruses were around for long periods of time and we just weren't catching them.
Yeah, I think that's a really good point, Brianne. And very possible. The sequencing is pretty spotty, as we saw, right? Mm-hmm. So as we think about not just maybe the source of Omicron from a human or a mouse or another, or other humans that we weren't thinking, but so first identified in South Africa, not because it was probably first emerging in South Africa, but they're doing really good surveillance there. Mm-hmm. The molecular epidemiology, the genomic epidemiology is really good, and so. They caught that thing. You could imagine it was circulating under the radar again, so to speak, in many other places, but that's sort of where it first picked up. And so, yeah, that's a really good point. And in general, actually, I think it's easy to, and for good reasons, to tout how remarkable it is that you at your fingertips now are millions of SARS-2 genomes, but that doesn't mean it's the perfect data set where it's evenly sampled throughout the world. It's quite a different story. Which can throw things a little out of focus when you're trying to ask these questions about origins.
The authors call this cryptic spread. Yeah, and I've been holding back on an observation that I heard about wanting to see confirmation and expansion, but suggesting that in some parts of Africa. There may be really high seropositivity in the absence of a lot of disease, okay? As if things might be different. Well, we should keep in mind things might be different in different environments. And so that would lend credibility to this idea of cryptic spread. Sorry, Vincent.
No, it's okay. Nels, the previous variants, did they fit well in the tree? None of them displayed this long branch as does Omicron?
Yeah, I'd say this one is pretty remarkable for that long branch. Phylogeneticists always are nervous about long branches, though, and so this gets exactly back to what Rich and Brianna are saying: is that you can get fooled where things that are actually really distantly related are more close, or even vice versa in some cases. So, yeah, it's an interesting question for sure. And I think in the end, maybe it's sort of kind of putting the punchline a little bit first is, we're not going to resolve this issue today, right? To definitively say the origins of this thing, which is kind of wild because fair enough, like a newly emerging when SARS-2 first showed up, also I think unfortunately pretty controversial still about those origins and how do we get to that even within the pandemic. Now that we have all of our attention, all our energy, our eyes focused on this thing, there's still these surprises that can come up and I think you're exactly right, Rich, that we're really kind of underpowered when we think about how this thing is moving kind of invisibly through communities around the world.
There is a little discussion in the introduction, which I wanted to just touch on because it's interesting. They make this point that patterns of mutations in RNA virus genomes can be dependent on the host environment. For example, certain enzymes, reactive oxygen species, cytidine deaminases, can make very specific changes. And even viruses that are quite different, and they compare polio, Ebola, SARS, can have similar spectra of mutations in the same host species, but then very different in different host species. Does that make sense, Nels?
Yeah, absolutely. And I think that's where that might be the strongest part of the analysis here is, so they do, a principal component analysis basically to ask that those sort of footprints or signatures. So are there certain mutations that are overrepresented and not mutations that even necessarily that natural selection is acting on, just that sort of cellular environment with all of these nucleic acid modifying enzymes, as you pointed out, the deanamases, et cetera. They're working a little bit differently, different flavors of them among different species, which probably is an echo of the diversity of immune responses, even in closely related species. But here it's like a nice kind of, almost like not quite a barcode, but just a little kind of hint at the types of mutations that are accumulating and how that might sort of point you could pick the cytoplasm based on those slight differences. And so, I think this is figure 3, where they show that the pre-outbreak Omicron mutations and how it clusters in a potential sort of swath with the mice. But let's be a little bit careful here. So, they draw these kind of ovals around to kind of draw our eye. But I would encourage our listeners to, if you're looking at this paper, and I think it's open access, but
it is.
Yeah, it is.
It's a kind of just for a minute, remove all of those ovals that they've drawn and just look at the dots for yourself and ask, wait a minute, are they kind of trying to, not to trick me, but are they definitely, making an interpretation of those dots? And so I think there maybe is a little hint of a pattern here, but I wouldn't kind of, bet the farm on this. I think there's some trend lines to some degree here, but it's a slightly weak signal.
Yeah, what I thought of when I looked at this is, for instance, the oval around the pig dots is huge. And to me, if they just did a connect the dots of each set of colored dots or something, maybe that would draw our eyes to something that it limits really what we could conclude, or it wouldn't make it quite so obvious. But that pig oval is huge. So.
Yeah, that's right. And so what they're proposing is that this thing started closer to the mouse oval. Now, if you look at it even a few months later, it's sort of back in the human oval, where you find some of the other variants, the betas, the deltas, the gammas, the alphas. So
since we jumped to this, let's talk about what they're doing. So they're looking at patterns of mutations of coronaviruses in different species, right?
That's right.
And this is based on the figure 2, where they're looking at the spectra, right? Like C to U, G to A to G, the whole spectra of changes, and how many you see in their different viruses that they're looking at, right? And so then the principal components is mapping them on a species level, essentially, right?
Yeah, kind of shared differences is what of trying to kind of take all of the data and sort of mash it together. What are the signals there? But so yeah, and sorry, I kind of did jump ahead there. I just got excited by your question. So, but if we go back to that figure 2, I think it's important to look at the y-axis, right, which is number of mutations here. Mm-hmm. And it's not like 500, 1,000 mutations, like where we're really, because I mean, this has happened again in this blink of an eye over the course of a couple of months. And so it's, between 1 and 5 mutations. And so that's the data's the data. But again, kind of when N is a small number here, I think that also sort of warrants taking a grain of salt before concluding, oh yeah, this guy was percolating in a mouse before it came and changed the world again.
So for the purposes of understanding this paper, can we define pre-outbreak and post-outbreak?
Yeah, let's go back to that. What viruses they were looking at. So if my understanding now is correct me, we have reference SARS-CoV-2, then they have 2B1.1 lineage, which they say we're close to Omicron. And then they have 48 Omicrons sampled before November 15th of last year, right? And is that what they call the pre-outbreak variants, right? Because before November, there was no Omicron outbreak, right? They were
so that's kind of the primordial Omicron. When Omicron showed up, they go looking around and they say, oh, we've seen this. Okay, and go back to the earliest one. Is that fair? Okay.
And then they say these 2 B1.1 variants, which were sampled April, May of 2020. So they're very close to Omicron. And they say this suggests that intermediate versions have gone undetected. And that's why we have such a long branch. So why would they be undetected? They just didn't do very well. They weren't as competitive as the other variants at the time.
Yeah, that's a great question. Sorry, Brianne, I think you were going to say something. Yeah,
I was just going to say, I could imagine 3 reasons. Nels can say if I'm correct here. But those 3 reasons are sort of similar to the hypotheses we had before. One reason is because they were in some other organism that we were not sampling. One reason is because they were in one particular individual who was not sampled, that immunocompromised individual. Or one reason is just because we missed them. Perhaps they were spreading in a country where we're not doing much sequencing, or some other reason why we just missed them. Is that right?
Yeah, that's exactly right. And said much more smoothly than I would have tried to stumble through it. So yeah, no, I agree completely. And here, so part or kind of a corollary, I guess, in a sense with that question is, what's the mutation rate here? So like, as we compare that to the sort of the core, or not core, but the other variants, the Delta variants, et cetera. And as you kind of trace along the branch lengths, how many mutations are on that long branch? And so I think that's part of the argument here as well, whichever of those 3 kind of scenarios that might be the case, is that there's a surprising amount of and this has kind of happened a couple of times, I'd say, with some of the other variants. There's this sort of moment where the genomic epidemiologists are a little bit surprised that this thing sort of has kind of launched a set of mutations. In a lot of cases, a lot of non-synonymous mutations, meaning that you're changing the DNA change is changing the amino acids, changing the shape of the protein, perhaps the spike protein, in a way that it's recognizing ACE2 of humans in a different way or of a different species. And the paper gets into that a little bit. I think that's the second sort of strong idea that they put forward for the mouse idea. But because if as you do those comparisons, you see a lot of mutations, so you can take kind of what was SARS-2 mutations as usual looking like. I think I might be, correct me if I'm wrong, if anyone has the number, it's something like half of a mutation per month.
Right.
So that's about right.
Sounds about right.
Yeah. And so here, I think it's, as you trace these lines on Omicron using the kind of calibrating pre and post outbreak by the definitions we were just talking about, it's like 3 times that. It's 1.5 mutations per month. Yeah. And so it's probably not that the mutation rate is any faster. The same enzymes, the same proofreading is all in place. But selection, there's these mutations, so many of them are non-synonymous. There's either from immune evasion or sort of host recognition or some of the other parameters, it looks like there's been a lot of action there. And so that's another, I think one of the authors would like to suggest that's another reason of sort of entertaining the idea of the mouse origin, is that if you move from humans to mouse, that you could imagine natural selection being pretty strong. There's a new playing field here. It's surrounded by slightly different proteins that the virus is interacting with as part of the replication cycle. It has to evade different immune defenses that the mouse deploys compared to the human. And so it gives a potential explanation for that seemingly increase in mutation numbers.
So they say that, right, so the mutation, the intrinsic mutation rate has not changed, but the selection is what is causing us to see more amino acid changes. So they use the ratio, right, of non-synonymous to synonymous mutations to look at that. Is that a fair way to look at that positive selection? I guess so, right? Now.
Oh, yeah, absolutely. That's kind of an old saw that we hit from time to time on TWiV as we're thinking about pointing out rapid evolution. And it's true. And so I guess, with this data, we'll do a quick knock on that case study I was hinting at. So if you just do sort of the average half a mutation, is it possible within humans that you could approach that sort of mutation rate? And so I think that case study, once we wrap up on this one, we'll have something to say about that.
So they calculate 26 of the 27 pre-outbreak changes in ORF-S. So it turns out that they identify 40 point mutations in the pre-outbreak Omicron, overrepresented in ORF, the S, the spike ORF, open reading frame, right? And so then they do a calculation of the DNDS with those, and they get 6.64, which they say is much greater than 1, which is, I don't know, what's that about, Nels?
Yeah, no, it's pretty wild. And so it's kind of interesting in the last couple months, there's been some conversations on population genetics Twitter, which might not be like your first avenue of social media to hang out with population geneticists, but it's pretty interesting. Sort of, as especially in kind of theoretical population genetics, the idea that you could have such strong selective pressure kind of acting over a short amount of time wasn't really in the cards for some of the the neutral theory, the near neutral theory, some of the older arguments about how kind of populations diversify or change over time under sort of all these scenarios. And so, but kind of, it's interesting to see, SARS-2 kind of teaching us or reminding us of how fast evolution can really move. And so, 40 mutations and with a DNDS ratio of 6, that's really something, actually.
They say that they look at all the other variants and they don't find anywhere near a DNDS of 6. Right.
Yeah. And I think there's some emerging structural work that's starting to, there's a lot of biology to understand here and how the spike protein is changing in its shape, how it's become more compact in the Omicron variant, and then how the surface has changed and some of the epistasis, how those mutations arriving together. Because there's mutations in there that just from some of our previous observations, you wouldn't have predicted that those would sort of be adaptive, actually. If anything, they might be deleterious. And yet maybe they're being tolerated or they're kind of taking on this enhanced action somehow in new combinations. And so that, and that could point to some unusual, so it's not just, I think what it points to, whether you believe it's a mouse or a different source, a human source, it points to this isn't just sort of person-to-person transmission as usual. There's another evolutionary step here that's happened, and then it's kind of come back to the human-to-human transmission. That's an important. I think that's a new observation. It's an important one.
Let me get this straight. This idea that the non-synonymous to synonymous ratio is so high implies that there's some really strong selection going on. Is that a fair statement?
Yeah. So the way this works is, and it kind of plays off the degeneracy of the genetic code. So you can have a mutation in a codon that ends up coding for the same amino acid, and that would be a synonymous change or a quote unquote silent change. And so the assumption is those kind of mutations, selection isn't going to favor or disfavor because it's just the same amino acid. And so if selection is acting on the level of the protein, then that gives you sort of a baseline or a denominator here to divide by the non-synonymous changes, which do change the amino acids. Turns out that's not a fair assumption. There is actually silent changes aren't so silent in a lot of cases. It's not just protein interactions that natural selection sees. It sees changes in expression. That could be outcome of not changing amino acid, but changing the sequence and how the proteins that interact with the DNA or the RNA influence the biological events that could be adaptive or non-adaptive. But it's a pretty good approximation, it turns out, and has been really useful for example, to focus on the host side. There's a lot of elevated DNDS of immune encoding genes or in receptors. So ACE2 has been rapidly evolving in bats. Not so much in humans, it turns out, or other primates, but some real solid signatures there. And so that's where, and I guess to kind of to calibrate this, the vast majority of genes, even in virus genomes, it turns out, have a very low DNDS ratio. There's more silent changes than there are non-synonymous because in general, so we have this sort of connotation of a mutation being negative, and that's because most are deleterious. You make a change to something you need to do genetically or biologically, and it makes it worse if you tinker with that. And so those types of mutations are said to be purified out of the population because any individual in a population that has one of these deleterious mutations just doesn't compete as well with other members or against infectious microbes, et cetera. And so that's most of biology, actually, is conservation. The reason we can pull up a budding yeast cell and learn about how our own cell cycle works is that our last common ancestors, we share this conservation over, in some cases, billions of years, a billion plus years. And then kind of in the middle zone, a DNDS of 1, where there's just as many non-synonymous substitutions as there are these quote-unquote silent substitutions. That would be a place of relaxed constraint. Selection isn't really looking at this in the sense that you're purifying out a negative thing or selecting for a positive thing. And then it's when you go above 1 that you get into these signatures of potentially adaptive evolution. And so when you're up by 6, and it's based on a somewhat small number. So I think the total of 40 mutations. The way the genetic code works, it's just by chance. There's about a 2-fold chance more that you'll get a non-synonymous. So you have to correct for that when you take this ratio of rates. The D part of this is the rate of the non-synonymous divided by the rate of the synonymous. And so that's what gets you then to this pretty strong signature for Omicron.
So I have an evolutionary question, and I may be understanding this completely wrong, and you can tell me that I'm out in space. But you mentioned that we're learning some interesting things about population genetics here. And I found that really interesting, that this is happening over a short period of time in a way that we didn't necessarily know. So if we are able to see this rapid change in such a short period of time, does that mean that we were maybe previously underestimating how much negative selection was going on?
Oh, that's a great question.
That we're finally now kind of able to see a situation where we see, oh my gosh, selection is way fast. We must have been ignoring some of that negative fastness before.
Yeah, no, that's exactly right, Brianne. So, and welcome to space. Great to join you out here. No, it's a really good question. And one way of saying it is, I've heard it phrased as the flaw of averages. If you're thinking about evolution kind of on the average, or the big principles for a population over 1,000 years, what's the average mutation? Or you're trying to derive these principles to kind of get at the, almost a physicist's view of the evolutionary process or something like that. Turns out that doesn't work so well. Evolution happens in these bursts, right? And so SARS-2, Omicron in particular, is showing us how bursty, in a sense, evolution can be. And, and that's going to echo on somehow for generations to come. And so yes, yeah, that by kind of melding these things together to try to define a principle, I think you can really fuzz things up and lose the real biology that's going on here. And so SARS-2, unfortunately, has been a great, just repeating reminder, just beating us on the head. Here's how evolution works. It's fast, and it can be drastic, and it can really be consequential.
So this diversification, this rapid positive selection after a virus moving to a new host, is typical. And so presumably it happened when the virus went from a bat to humans or whatever, right? But we just missed all that, correct?
Yeah. So that's really interesting, right? Is like, and a reason that, there are many reasons, but one, from a kind of evolutionary genetics perspective, is you'd love to continue sampling to get those viruses that would, the closest relatives to the outbreak, right? Because then you can kind of step through that process much more carefully. The difficulty now is, I think, last time I checked, I think the closest related, SARS related coronavirus was 95% identical or something like that. That's for a 30 kb RNA genome, that's a massive amount of mutation. So you can't really follow or trace what were those steps that happened kind of late 2019. It's a fascinating question. You'd love to see it. But Omicron may, if the mouse hypothesis holds up and going back and forth, it might provide sort of a little insight into that, and that would be consistent with this scenario. Again, it's even hard, so are we going to find, if let's just assume for a moment that the mouse origin is true. Are we going to find that mouse? I don't think so. Where is that mouse, and how many mice are just hanging around? It's really hard to do.
It's funny. I was talking about this on the train this morning with a guy next to me, and because I was reading this paper and he asked me questions. He said, "Okay, what if I sent you from all over the world, 5 million mice?
Yeah.
Would that do it? And how much would it cost?" He said, "What if I could get them from all over the place?" Because they say, who knows where to look, right? We don't know. And there are a lot of mice. Mice are 40% of mammals. It's a lot of mice out there.
Exactly.
So the likelihood we're going to find it, I think, is pretty low, right?
And mice in the wild are going to have a pretty short lifespan.
Yeah.
That's right. And are the mice, if let's say SARS-2 is moving through them, are they suffering? We don't have mouse clinics where we diagnose respiratory infections. And so, this is invisible. Most of virology is invisible and vast.
I mean, what it tells us is something that Rich and I heard many times on our travels. We need to be sampling way more than we are, because you can't do it once the outbreak is here. You can't go back. As Kathy said, the mice don't live forever. But if you sampled continuously and knew what was out in certain populations, maybe it would help. But we don't do anything near that, right?
Yeah, that's right.
So they make this interesting point. They say most of these spike changes are in the receptor binding domain, right? And if they compare it to other variants and viruses from 3 chronically infected patients, they don't see such an overrepresentation in the spike or the RBD. Is that correct?
Oh, yeah. So we're moving to this case study?
No, I was just there. It's in this paper. They reference 3, and they just say, we did not observe such a level of overrepresentation of mutations in spike or RBD as in pre-outbreak Omicron. So it's quite distinct, right?
That's right. So, and that's, kind of taking that half a mutation per month as usual sort of view as you're looking at human-to-human transmission mostly. This is different. Yeah, absolutely. Yep.
All right. That's it. That's just mentioned here, and I wanted to.
No, it's a good point.
But we'll bring it up again when we get to the
exactly.
They've got a couple in their figure 1. They've got a couple of points on their charts in CD and E where they compare variants of concern and isolates from chronically infected patients and compare that to Omicron and basically the isolates from chronically infected patients don't show any special characteristics.
Yeah,
that's right.
And even before that, in 1B, where they have this lineup of all these different mutations and virus isolates, and you can see the RBD in the very top one, the pre-outbreak Omicron. It's this huge peak, and then the others, nothing much.
Yep, agreed. And so this actually speaks again, I think, to sampling and how even when you have what you think is good sampling, just that vastness of biology all around us, they're like, hold on another minute, there might be another example to look at. and that's what we'll echo on here in a minute. But that's right. So that also kind of points to because they're in this sample of chronic patients and seeing a smaller amount of mutations, that's sort of pointing them to the the more exotic reverse zoonosis, return zoonosis situation. And then there's more evidence too, right? So they also then they don't. So what you'd love to do is some assays, right, to express that spike. So they're looking at ACE2 binding. And so they use this program Haddock. And these are getting this kind of in silico analysis getting better and better, and allows you to predict how, based on what we know from in vitro assays, binding assays, et cetera, to predict, given this set of mutations, how well is this going to bind a certain flavor or variant of ACE2, the host receptor for the spike protein. And that's where they point out as well, the Omicron receptor binding domain starts to, the score, the in silico score matches best, or is an increased score for mouse versus any of the others that they've sampled here, and some of the mutations if you just take them in isolation.
This kind of analysis reminds me of a paper we did a long time ago, and I'm sure you remember. There was a PNAS paper where they did modeling of the ACE2 spike interface of various animals, and then they said, okay, this will tell us which animals are at the highest risk for being infected. And at the very bottom of the list, I believe, was mink. Because their analysis said not likely to be. And I just take this with a big grain of salt. I just don't think, first of all, the binding is important, but it's not the only factor, right? Everyone seems to say, this controls host range, right? Receptor interaction. Well, it's one of the controls, right? But there's others. Once you get in the cell, there's things you have to do there as well to reproduce. So this ignores all that. And so the combination of those two makes me very suspicious of this kind of analysis. I think some of the other things they do in this paper are far more convincing than this. Is that a fair criticism, you think?
Absolutely. Yeah, I agree. And call me old-fashioned too, but yeah, I'd like to see some data, some get out the pipetters.
Well,
and let's
wet experiments. Yeah, they would argue this is data. Right.
Oh, yeah, good point. So, and the other limitation here, right, is that they, at least for the profile they show us, they've made a specific choice. It's like mouse. And let's run this against mouse and see how these go. What about rat or mink? What would that profile look like? And would it be the same as this actually? Like, would Omicron do better with those ones or bat? So I agree that I think it feels a little bit cherry picked, and it's based on a limited set of information as well.
This is a good time to sort of explicitly state that this entire paper is computational biology. Okay, nobody's pipetting anything.
And that's okay. But there's like the mixing of the two can be a really important way forward. And definitely, to guide some ideas or hypotheses, but you really want some evidence.
Yeah, I think making the protein and studying some of it would help, right? It's not the whole story, but this is going to be hard to do because you have a lot of changes. And you don't want to just look at them one by one because, as you were saying earlier, they can have interactions, right? So that becomes how many combinations are you going to make? And the problem is that people will make a few changes and go, oh, this recapitulates everything, and that's it. But it may not be, right? So
it's kind of I agree. It's hard, and it also, it kind of illustrates like science at the pace of a pandemic, where we've put such a premium on kind of trying to discern new information, and that's totally natural and makes sense. But I think that this is a trade-off of it, is that we have all of these rough drafts that we then put forward and talk about, and the revision process takes a while. And so I think, science outside of a pandemic, we kind of do that revision process a little less publicly. We might, show some data like this to some colleagues who then might, run an assay or two, and then you might put it on as a preprint on bioRxiv, and other people will talk about it, give you feedback. You might revise it and then launch it for peer review, something like that. And that process can take years, but you end up with, it's still a rough draft, but it's a much more refined one. You've already made a few revisions before you've just put it out there. Of course, in a pandemic, we want to get as many eyeballs on this, get as fast an understanding as we can because of the public health consequences. But it's important to kind of keep that perspective and understand. I think that's been, honestly, part of our current challenges with bringing the public along on, understanding the science, where we get these comments about how, well, you told us to do this time and told us to do this other time. And I think that's a little bit of an outcome of where we're seeing this rough draft style science. And so at a different pitch. And so how do we kind of explain that? It's a new challenge that we've created and has, I think, been a massive challenge for public health and just getting some buy-in as a community that there's some real advantages to working through this together and not sort of launching these flame wars against science and the scientific approach. Especially explain that in the sort of cultural environment of changing your mind is naughty, Yeah, that's right.
Mm-hmm.
Yep.
Indeed. No, I'm thinking of the accelerated mutation rate, of Omicron. It's been covered in the press. Omicron is mutating faster, which is not right, as we have said, but it's a nuance that's hard to understand, right?
Yep.
So I kind of want to come back to this set of different experiments that they do, because it's kind of asking, like, what's your favorite child? But, when I saw figure 2, I found that fairly convincing. But then Nels has just pointed out that for the pre-outbreak y-axis for part B, the n is small. Yeah. Okay, so now it's become a less favorite child. But and we kind of already dissed on figure 3. But I'm just wondering, taken together, does that help build this case, or how do you feel about that?
Yeah, great point, Kathy. And I think that's especially when you're doing in silico analysis or making inferences with evolutionary analysis, you're always looking for that kind of orthogonal. How do you attack a question orthogonally? How do you hit it with complementary evidence? Because then it becomes, it starts to kind of calibrate or triangulate on a pattern or it lends more support to the idea that you're putting out there. And so I think that's exactly the right approach. But then, I don't think we're, as we're going through and sort of not dissing on it, but like critically evaluating or analyzing it, then we have to weigh, like, okay, well, how valuable is that sort of piece of evidence as it's presented, as it relates to the other ones for the bigger picture? And yeah. So a correlate question for you, Nels, is that as you were reading this, did you think to yourself at any point, well, did they look at this? Okay, because in taking an orthogonal approach, there's the potential for trying something and it doesn't support your hypothesis and saying, well, we won't talk about that. So did you think, they should have done this? No, great. Yeah. And I think that's kind of, it's a great point, Rich. It sort of like peels back the scientific process a little bit, right? As we're readers or, advancing these ideas, is to always do that kind of self-calibration. Is like, well, you might be making you want to make a strong statement about the idea, but, are you weighing that with the with the information that you have, and kind of balancing that, so, you don't want to be, if you're totally wishy-washy, here's all of this evidence and it could mean anything. That's not the most, you're not going to be talking about that on TWiV, for example. And so, but obviously, and especially as it gets interpreted by, folks in different fields or the general public, et cetera, it can also go too far, get interpreted, even the data that you're presenting, in a way that you're not weighing or hitting that. And then that's the question is, are they sort of missing out on other points or even they did some other analysis that didn't quite fit into that picture. It didn't kind of hit that orthogonal thing to support the idea, in this case, the mouse origin. And does that get sort of then pushed to the edge? There's a danger of that. And I think that's part of the process of going through this, is to, I think, try to be as open-minded and to weigh things in as reasonable a way as possible. Easier said than done. But reading this, nothing leapt to your mind that they should have done this or that? I think you've got something in mind. No, Richard. I don't. Yeah. Oh, you don't? No. So I didn't have that impression. So actually, in the way I read this paper is kind of maybe another sort of like symptom of pandemic science is. So I first read about this paper on Twitter with people interpreting it and the points kind of going point to point. And a little bit of a big, like, in addition to this manuscript, some other data. I mean, other people, other groups, of course. I'm sure there's actually multiple groups who are picking up pipetters now, taking the Omicron set of mutations, doing, and already I think we've seen crystal structures, and then it's, right behind that will be some of the assays, and we might see a panel of ACE2 from rodents, minks, et cetera, to try to see what's going on and get stronger data in ways that just take more time. But anyway, so that conversation has been also happening on Twitter among sort of a group of folks who do this for a living, as they're the genomic epidemiologists and others, evolutionary virologists, others. And it's been interesting. So they've kind of been floating these exact 3 hypotheses based on sort of real-time data as it comes on, including this kind of stuff. And so I think it has broken into 2 camps, sort of the mouse origin camp and then the human origin camp. Because this is all kind of late breaking, it's sort of like, there's almost, and as we've, I think, hopefully set out, there's nothing really definitive here. And so this is also maybe another cautionary tale is that, as scientists, we aren't robots. We're also kind of working on our intuition, or we might pick up a pet hypothesis and sort of see, does the data match that view? And so I kind of approach this somehow with thinking, like, I just don't quite buy the mouse situation. It could be right, but my intuition points me towards this is all happening in humans. It just seems like a more parsimonious, sort of a simple pathway. Now that's, if we look as Kathy's pointing out, as if we look at figure 2 and, some of the patients from these immunocompromised, and the difference that they're drawing, or the between the number of mutations, et cetera, that kind of works against my intuition in that case. But there's more going on here, and so that's what got me onto this case report. So this is maybe is it okay for me to step forward onto that? Yeah.
The I guess one piece I was just going to say is I think to me, I look at this and I feel like, in some ways, almost 2 things are getting conflated in terms of, is it reverse zoonosis and is it specifically mouse?
Yeah, that's
right.
And
how much does this paper support, not human versus mouse specifically, as different options? And I think that the paper that you're getting ready to switch gears to sort of hits the no, it's human side as opposed to the no, it's not human side.
Yeah, that's right. And so, no, it's a great point, Brianne, which is, so I think reverse zoonosis, you might want to be a little bit open-minded about that. It's like, why do we immediately go to mouse? I mean, there are some reasons, of course, like just human contact with mouse, although I'd put rats into the running there. I'd love to see that haddock analysis with a rat, I guess.
Yeah.
What about the part where they look at all the mouse-adapted viruses and they make comparisons between the amino acid changes in those? Do you think that strengthens their argument at all?
Absolutely. I think it's, so it's consistent. It's those low numbers. I'm trying to remember. So so mouse, we've got mouse is kind of intersecting with bats, cats, and dogs. We have camels, cattle, pigs. I actually don't see rats on here.
Yeah, I don't see rats, and I also don't see hamsters, and I know there have been some hamster reports out there too. Yeah,
we don't have mink on here as well. I think the point is really well taken, Brianne, is like, evidence of reverse zoonosis could fit into the mouse idea with some sort of circumstantial evidence, but I don't know, that's sort of a second leap to take here in a sense. And then from an argument, maybe of parsimony, we're taking, and this is difficult too, because all of these events that we're talking about are super rare, right? This Omicron has hit the jackpot when it comes to competing against the Delta variant, competing against any other SARS-2 variant, and making this almost just massively dramatic sweep through the global human population in the course of a couple of months. Really wild. So I kind of favor, at least my intuition sort of points to me, well, it's a lot easier to just keep hanging out in humans and continuing to infect them. It's already made that spillover event in 2019. That feels like, from an evolutionary standpoint, a pretty massive step. And so, and this finally gets me onto this case study that I pasted in here. So this just came on my radar a couple days ago. This is from South Africa. Some of the same epidemiologists and related public health scientists who were pretty heroic, I have to say, in identifying Omicron and getting it out to the world really quickly with some negative consequences for doing that. And, giving us a head start on look what might be coming. And with something that can spread across the globe in a couple of months, having even a few days can make a real difference. Whether we take advantage of that time that was bought by sharing this information early and often is another question, but that's an important part of the conversation here. So what this case study does is it actually looks at a immunocompromised patient. I think it's a young woman in her early 20s who was born with an HIV infection and then has had a sort of a mixed history of keeping up on her antiretroviral drugs. Sort of in the course of the case study, became infected with SARS-2 and didn't control it. And so the infection went for about 9 months before they kind of got on top of it with sort of reinstituting her HIV therapy and then also treating the SARS-2 infection as well, and I think resolving it. But in that 9 months, some interesting evolution happened, sort of in contrast to those other immunocompromised cases that were put up in the mouse origin paper that we just covered. And so it was sort of by luck, it seems like. So they had just sampled at that first, she shows up in the clinic kind of at month 0, when she was probably first infected or near the infection with SARS-2.
Which variant did she have, Nels?
I think it was the Alpha variant.
Okay.
It wasn't the Delta variant. It wasn't Omicron. And there's no evidence that this variant, so it underwent this sort of evolutionary course during her infection, but never transmitted to another human. But the fact that they had that early sample and were able to recover basically the whole virus genome, and then this sample at the end at 9 months, they could just do a simple comparison. And so what they saw over that 9 months was 20 mutations from beginning to end, or 22 mutations, and 20 of them are non-synonymous. And so now all of a sudden that, 0.4 mutations per month is looking a lot closer to the 1.5 per month. All of a sudden the DNDS ratio of 6 that we were just sort of touting as this rapid evolution, that probably weighs in at about DNDS of 5 for these 22 mutations. So it's right, all of a sudden, we're right back with a situation where it's possible that an immunocompromised human, so not this patient, but another patient or a set of them were what kind of led to the origins of Omicron. And then the sort of advantage, or not advantage, but sort of maybe the intuition of this hypothesis is you don't have to do this fancy spillover event and then spill back. You can just can kind of percolate within a human, and then it can just sort of reemerge with a new set of mutations.
So Nels, do they look at the types of the mutational spectrum, for example, and compare it to the others as they did in the paper we just went through?
Yeah, not that way, but they do. And this actually gets to a point that you made a little bit before, Vincent, was, so all of one of the surprising things about Omicron, right, is that sort of concentration of non-synonymous mutations in the S gene, in the spike. Mm-hmm. Here in this patient, like of the 22, about half of them are in the spike. The other ones, there's a lot of mutations, non-synonymous mutations in ORF1. And I think one in ORF8 as well. So that's a little bit different than Omicron. But again, just given the sort of small numbers here, maybe to me, the bigger impact of this observation is just that you can really kind of hit those those parameters that you would wouldn't be surprising that Omicron could have that.
And there's certainly numerous immunosuppressed people in the world, many of which have not had medical care and have not had isolates taken. So who knows, right? That's
exactly right. And to me, this really, I think there's a message in here, a bigger message, so it could, and I'm not going to try to say that this definitively says this is coming through immunocompromised humans. It could be the case. It could be from a mouse or another, as Brienne's pointing out, from another source, another animal, a reverse zoonosis and zoonosis, or a combo. So by the way, there's not just one Omicron, right? There's at least 3. BA.1, I think it's BA.2. So BA.1, I think, is the one that really made that first wave. And now we're seeing this echo of BA.2. And it's not that this is sort of a derived version of the first Omicron variant. I think this has 20 mutations compared to that, and probably emerged at about the same time. And anyone who, if I'm getting this wrong, please jump in and correct me.
I'm not super up to date on it.
Okay, so I think that's the case, and so, and it's a little bit, not to like try to throw gas on the fire here. So the good news, right, is that it looks like at a lot of locations, the cases of Omicron are coming down, and that's still the case with this BA.2, the second Omicron, but it's sort of, it's replacing, as these numbers fall, it's replacing that first one. It's outcompeting it. So even as things get better, at least in the short term, there's still this evolution going on. It looks like these are competing against each other somehow. And so now I think, at least in places like the UK, maybe Denmark, other places where there's good sampling again, this second variant has taken over, and it's actually quite a bit different. Like it's still called Omicron, but that's kind of a fine line. It's sort of, you could almost imagine giving it another name if it really becomes consequential.
But they have a common ancestor. Is that correct?
Well, so that's it's hard. So they definitely, so they cluster together. I'm going to be a little kind of evasive on that answer. They cluster together as if that's possible, but it's also possible, for example, that these are independent emergences from different immunocompromised humans, or maybe one animal and one human, or something like that, that were kind of close to each other at the same time. And so it does look like Omicron in the sense that, so it's very different than, there's a reason we called Omicron and not Delta version 2.0 or whatever, because it was at a really different spot. These are sort of together on the tree, but what that means for a common ancestor is a little more murky.
Go ahead.
No, go ahead.
Where was BA.2 first detected? That's all I want to know.
I think it was, I don't know, actually.
It was in South
question.
Africa, right?
I think I think this is all coincident. Yeah.
Okay.
I just wanted to come back to something you alluded to, that maybe with this case study, there's been some fallout in terms of maybe because it's from South Africa or something. If you could just enlighten us just a little bit.
No, thanks, Kathy. Yeah, so I was kind of starting on this point, which I think is, I think regardless of the details of the evolution here, there is a really important public health message, which is, where are we in our global distribution of vaccines? So as we've said kind of throughout this, right, what are our best tools as hosts for SARS-2 to counteract this? It's still the vaccines. And what have we done with these vaccines? We haven't distributed them evenly. And so when we take that combination, I think and again, whether or not Omicron, what the origin is, because we're seeing these evolutionary patterns that are consistent with exactly a new variant like this in an immunocompromised patient anywhere across the world, but in particular in places where vaccines aren't distributed, we're just sort of setting up that uncontrolled experiment again and again and again. And so we're giving the virus every chance to do this. We're also doing this in very resource-rich places. My neighborhood in Utah, a lot of people who are refusing to get vaccinated. A lot of people who are immunocompromised have autoimmune situations. And so I think, one of the real advances, the miracle of medicine has been to have much better outcomes, healthy lives as immunocompromised people. One of the challenges is then, I think for me, it highlights why we really have to work as a community to support each other if we want to get past this thing, and not to have to, wear out the Greek alphabet with more and more variants as this thing continues to sample mutations and come out and surprise us in ways that we hopefully are starting to anticipate. And there's some clear steps that we could actually take.
A little sidebar about this persistent infection paper that interested me. Following on your comment that they, basically found this more or less by chance. And it's not quite chance. So there were 3 samples, as I understand it, one at what we'll call time equals 0, one at 8 months, and one at 9 months. Okay. And the first one, the 8-month sample was the first one that they looked at. And that just happened to be sequenced, but that's because they have a sequence surveillance thing going on. And that particular sample fell into that surveillance. And they said, oh, okay, so this is interesting. And that focused their attention on it so that a month later, they actually did, consciously did the sequence and compared them. And they say, oh, well, that's really interesting. And then they looked back and saw that they could go back into sample archives, and they had a sample of this person from 9 months previous, and they could sequence that one. They have this comment in the discussion where they say the case furthermore highlights the value of well-coordinated and thoroughly established genomic surveillance efforts. Fortuitously, the September sample from this patient was sequenced as a part of the NGS-SA effort. It was flagged as warranting further investigation as a beta variant, which by that stage had become very rare by the sequencing and sequence analysis teams who contacted the diagnostic virologist and those the requesting clinician. Good connections between sequencing laboratories, routine diagnostic laboratories, and frontline clinicians are indispensable to identify and investigate such cases. So I'm thinking Pasteur says that chance favors the prepared mind.
Yeah.
I would say this says that chance favors the prepared community.
Yeah.
Riffing on your community thing.
And to go further from what Rich is saying, I don't know if you guys noticed this, but in between the earliest sample and the 8-9 month later sample, the patient moved across South Africa. So the first sample was done in one province, KwaZulu-Natal. The later ones were done in Western Cape, which are basically the opposite coasts of South Africa. KwaZulu-Natal happens to be a place where there are lots of HIV clinics and there's a lot of HIV study. And then Stellenbosch is another great area for biomedical research. And so it's sort of fortuitous that this patient moved from one place where they do great sequencing to another place where they do great sequencing. And all of this could be followed. You could imagine there are similar things in lots of patients that are not in places where they're sequencing in the same way that they are. And where you can't actually record what happened to you on one coast of South Africa versus the other.
Sure.
Yeah. Really good point.
I also want to go back and riff on something you were talking about earlier. This is really big science, okay? You talked about, part of your conversation earlier was essentially about how scientists formulate their own biases and about how we try and deal with that as scientists. And especially late in my career when I became more and more familiar with how my mind worked, okay? I found this fascinating. I've talked about this before, but I want to talk about it again because it fascinates me. When you construct an experiment or you're doing a series of experiments, you are likely to construct hypotheses about what's going on that you really get wed to. Like, this is the truth. And then you do an experiment and the data comes out and you look at this experiment and it says right in your face, Condit, you're dead wrong. And there's no way around it. You're wrong. And then comes the moment that fascinates me where I have to look in my head and I can feel my biases sort of struggle and then unwind. And then I have to kind of start over. And then, of course, I just rebuild a new story, okay, that could be wrong as well. But that head game is one of the most interesting things going on to me. And it's really challenging. Okay. Yeah. It's challenging to accept that you're dead wrong and disassemble that story and reassemble a new one.
Yeah, no, you're totally right. And in fact, many articles have been written about this. It's really very difficult for many scientists to admit that they're wrong, in fact. And so they stick to hypotheses, grant after grant, and never say we're wrong because they're afraid they won't get their money next time. I think it's a real issue, and it's good that you were able to recognize it, I think.
It's a huge issue too, and that's how science goes forward in the end, right? Yeah, how we get a new, and so I mean, another way of looking at it that I try to convince trainees in my group, for example, is to say, let's falsify this. Let's get evidence one way or the other as quickly as possible. Let's not find evidence that proves your idea. Let's find evidence that just allows us to make progress. And the best case scenario is that you get a definitive answer so that you can do exactly what Rich is describing. Let your two sides of your brain or whatever wrestle through the through the revision that has to happen to refine that. That's what's going on inside the scientific brain, I would say. And just to touch quickly on, Brianne, your point about following the patient over, and I think this, maybe this gets at another bias that scientists have from time to time, which is that, the science is important. And so people should pause their lives and, allow us to collect the data somehow that's going to be the best for this study. And that's not how people live. And so if you're really going to do this kind of surveillance, it has to go to how people live their lives. And we'll do that regardless of what scientists are doing. And that's how you actually get better information. You get better data, is not to try to, we're not going to bring humans like they're lab mice into, right? It's the other way around, where we have to follow each other in the wild, so to speak. So that's an important point. I
also,
agree
I
with
think that personalities are part of this mix, right? There's some very forceful scientists with big labs, a lot of money, track record, who people can't challenge them, right? And sometimes it takes new person coming into the field to change things because you have such inertia from the status quo, right?
Yeah, agreed. That reminds me of your colleague's book, Stuart Firestein's book, Failure.
Yes.
Why Science is So Successful.
Yep.
And he just lays out a couple stories like this, how scientific progress can be delayed, not just like a year or two over a grant renewal for generations, depending on the purported authority of that sort of famous person. It's really a detriment to the scientific process. Yeah.
I wanted to go back to one last point here. In the discussion of the first paper, they make this point that their docking predictions they use the word show, that adaptation of Omicron to mice also promote its adaptation to other species. All right, so I don't think you can make that statement. Just because on that simulation it looks good for a camel at the same time as it doesn't mean that it actually is, all right? So I think it should have been a little less strong. However, it's not the main point I want to make. They say, if that's correct, then once a variant acquires a new animal reservoir, it can accumulate mutations which allow it then to infect another animal, right? If they're right about this docking simulation. They call this chain reaction of host jumping, and they say, consistent with this possibility, numerous mutations were identified in the spike protein gene of SARS-CoV-2, amplified from wastewater samples. And they reference a preprint by Davida Smith, Monica Trujillo, and others who were on TWiV actually to talk about their wastewater sampling in New York. And the paper is tracking cryptic SARS-CoV-2 lineages in New York City wastewater. And so this is all about them finding lineages that you never see in people in wastewater. And the idea being maybe they came from animals. Anyway, one of the authors, John, where is he here? John Dennehy, the senior author, he wrote me, he said, this paper is now accepted. Would you like us to come and talk about it? So I think based on today's conversation, let him come on and talk about this. I think that's quite interesting. It's just fragments of RNA, right? They do little PCR, but it's unique. They're clinically unique, right? Rarely observed in clinical samples. So including some of these changes that we talked about today in the receptor binding domain.
Yep. And that kind of gets me thinking about, forecasting here, which is nearly impossible, of course, but where's SARS-3 coming from in a decade from now or something like that?
Oh, God, I don't even want to think about it.
And, a fair bet would be from bats. And what's going on with ACE2, the receptor? So getting at that point that you're making, Vincent, about, these viruses getting into a host that has a different or variants of that receptor and then sort of sampling mutations and tinkering. We see a lot of adaptive evolution in bats for ACE2. And so what that means for spillover potential, or how these things are kind of testing that through random mutation and selection, I haven't seen anything that definitively, makes a lot of progress on that. But I think that's an area where I would point to try to get a handle on this. And there, I think the advantage you have with the bats is sort of to actually see rapid changes in the receptor, even among closely related species. And so it's sort of like the best approximation of a natural experiment where, by and large, the rest of the interactions are relatively the same compared to if you're going from mouse to human or mouse to bat, where there's a massive evolutionary gulf there. It's really hard to kind of to pinpoint the impact of these changes, but because there are closely related bat species that have different ACE2s, that's sort of the, if we think about this from that evolutionary perspective of nature doing the experiments for us, and then us listening in or trying to get information from that seems to me like the best place to look. And by the way, that's most likely where these SARS-related coronaviruses are hanging out and spilling over.
So is the driver of that ACE2 diversification, do you think those are infectious agents in bats?
That would be my best guess, yeah. And so again, we don't have bat clinicians out there checking if bats have common colds versus more severe respiratory situations. And so that also, there is this entire and interesting, I think, idea that bats have sort of, interesting immune systems or sort of super immune systems. I'm still waiting for the bat clinicians before I buy into that.
They have many copies of ApoBec genes, bats, more than any other animal that we've looked at, right?
Absolutely. Super interesting to me. That makes me think that there's been a history of some pretty severe infections that have wiped out a lot of ancient bat populations. And we can even find, cases where bats are struggling today, right? White nose syndrome. It's a fungus, not a virus, but that's now wiping out populations. That's like evolution happening before your eyes. Hopefully not extinction events, but the bats that remain or become, the ones that are resistant to white nose or other infections, those will be the ones that are sort of repopulating bat habitats.
And as you said earlier, these are episodic events, right? It's not chronic. And it reminds me of a paper we did on TWiV where they talked about there was a period in history when there was a lot of introns going into some genome that it just was a short burst of intron acquisition, and then it stopped, right?
Yep. Yeah.
So we're going to have listeners who I'm sure are saying to themselves, okay, gang, after all of this blah, 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 where did Omicron come from? Okay. And I think the answer is we don't know. Is that right?
Yep.
No, we don't
That's
know.
right.
These are some ideas. These are some of the data that can be used in support of those ideas, but we don't know.
I would. Yeah. Oh, sorry. Go ahead, Vincent.
They say their last sentence is these data. Well, they're very convinced it came from mice, but aside from that, we need to have more viral surveillance and sequencing in animals, especially those in contact with humans. And I think that's really the outcome of this work, right?
Yeah. And I do think, as Brianne was pointing out, sort of that third hypothesis of sort of regular person-to-person transmission, that feels a lot less likely, that there is something going on here, whether it's in a different species or a immunocompromised human, that's the more, between those, that's the more likely scenario.
I mean, you should point out that, all the species on that principal components analysis are the ones they looked at, right?
Yeah.
There are others they didn't look at. Could be there too. So I think probably not going to ever know, right, where this came from. But maybe the next one will be better at. I just don't know. But it's quite interesting. The problem is, as we've mentioned already, the press is in this because it's a pandemic, and so they want to know, did it come from a mess? And I'm almost like, well, why? Why do you want to know that for sure? We have to just get vaccinated. Doesn't matter where it came from.
Let's vaccinate the world and do more surveillance. How about that is our answer.
Well, and there's, I think my hope is that kind of, so that if we avoid that kind of ripped from the headlines science, and I'm encouraged that there's a lot of groups who are really taking advantage of this incredible data set that builds, more and more samples by the day. There are real advances hiding in those comparisons, hiding in this data, where, something that we learn about in the next year or two by doing a more careful or sort of behind the scenes analysis could provide insights that will help a decade from now. And if that's true, that kind of just echoes on the kind of, the thing we talk about a lot is the value of basic science. So why is it that we could deploy these incredible vaccines so quickly? I mean, really a moon landing kind of situation in my mind for deploying these in a 1 year's time. Incredibly, some combination of skill, preparedness, luck, all of the above to point that out, or, to pull that off. And then, which to me also stands in sharp contrast with our ability to distribute this and or take advantage of this if it's available. But it was those discoveries 10, 20 years ago, in a lot of cases that we didn't even think might apply to something like this, that really came through to make that possible. And so the question becomes, are we doing that today in ways that we won't know it'll be worthwhile a decade from now? And so, that's all my kind of common refrain for let's keep doing basic science. Let's keep investing in it.
Well, look at how much trouble Kariko had to do her work, right?
Yeah, absolutely.
She could not get it funded. And that's the foundation for this, for making these mRNA vaccines, right? Because people don't believe it. This is a fundamental flaw with the funding system. We did this last time, so I don't want to do it again. But it's ridiculous that she had trouble getting this work funded. It was clearly important. It's modification of bases. Yeah, let's find out what it does. So that's the problem, Nels. I'm not sure that we do everything that we need to be doing because there's too much translational aspects of science. Anyway, can we do one round of email? Because we have a whole bunch, and these are all pretty easy. They don't ask us any tough questions. So, Rich, can you take that first one, please?
Sure. Dora writes, I live in Austin, Texas. But even when I was growing up in upstate New York, by the way, Dora, I spent 12 years in Buffalo, and Brianne is from upstate New York. My father used to tell me that the great threat to orange growing in Florida was not freezing weather. It is warm weather that comes before they're done with the freezing weather. So this is in response to a conversation that we had a couple of episodes ago about about the impact of cold weather on citrus, precipitated by Dixon. The citrus trees bloom and then the blooms freeze. In Austin, I can readily see what the problem is. When I moved here in 2000, there was a reliable pattern. Temperatures used to last rise to the mid-80s in early November and first rise to the middle 80s on Valentine's Day. On Valentine's Day, you go down the street and suddenly there are leaves and flowers on the trees. With ash trees, it's flowers, and on the ground, flowers are blooming. But in late winter, early spring in central Texas, the weather is very volatile. Very warm weather and frosts alternate on a weekly basis for the next 3 or 4 weeks until the last frost in March. Tell me about it. Since I have been here, the date of the last frost has subtly gotten later as the weather gets less stable. If a killing frost where the temperature falls into the 20s and stays there for a few hours happens, after the leaves and flowers appear on the trees, they die, and they don't reappear until April, leading to a depressing early spring. Yes, that used to happen more times than not. Last winter, it stayed so warm that the flowers never stopped blooming. Then suddenly on Valentine's Day, the polar vortex, which Texas rarely sees, came spinning south and got cut off and sat there for a week. Temperatures that never get below 19 in Austin plummeted to 5 degrees, while storms that normally hit Washington and Oregon went around the big polar vortex and came right over Austin every other day. Our infrastructure isn't designed for this, and many people had no heat for an entire week, and everyone else saw it keep going on and off. All over the city, water mains broke. Trees had broken out in leaves and flowers a week or two early, and every single tree branch in the city that had flowers, leaves or flowers on it died, all caps, leading to dead trees and trees with half their branches dead, usually on the outside of the tree. Many bushes and trees died or died back to ground level. The polar vortex, of course, is breaking down because the Arctic is warming and can be expected to spin south regularly. In fact, this has been happening for years. It just doesn't usually head for Texas. This year, it stayed warm so late the last leaves fell off the trees this weekend. That normally happens by Christmas Day when everyone goes out and rakes up their leaf. Yours, Dora. Well, she's singing my song, you know. I lived through all of that. We lost plants that normally survive, and we're still recovering. And next week, after 65 today, a couple of days from now, it's going to be crazy cold.
Weird stuff. Now, I thought of the possibility that Dora is actually someone who hates the weather, and she wrote a long letter about it just to make us. But probably not.
I don't think so. I think Dora's into the weather.
Brianne, can you take the next one?
Sure. Racaniello writes, Dear TWiV, I thank my lucky stars that you exist and always tune in with pleasure. I am an unconditional fan. Unfortunately, I can't say the same about doctors these days, and that is a strange feeling. Just generally, how are we, the wider public, to understand some doctors' reactions in the UK and elsewhere in saying that they themselves should not have the vaccine? I am less angry than puzzled. I am fully vaccinated, booster included. Such people say that they are grateful for the vaccines, yet they themselves will not take it. In the UK, but only in England, about 10% of medical staff will be affected by the government decision to make vaccination compulsory for medical staff. I am not aware that you would have referenced to this, but if you have, I would be grateful if you could point me to the podcast in which you have discussed this. I would be equally grateful if you could discuss this from the point of view of how we are to understand and relate to science if messages become so nuanced as they have become. I always thought that even if they are researchers, doctors working in a clinical environment know more about science than I do, being, as I am, educated in sociology and the humanities. Thank you for your efforts. TWiV is beautiful. Kind regards, Graziella. I don't want to say super negative things about doctors, so I'm going to keep my mouth shut.
They need to get vaccinated, Brianne, don't they?
Yes, they need to get vaccinated.
I mean, they are taking care of your health. That's their job, right? Unless they have a medical reason not to be vaccinated, but 10% seems like too high. I don't understand this either. This is not something that, Graziella, we would understand or even talk about other than say that get vaccinated. And I don't really get why. I mean, many other people have similar positions. I don't get it when doctors do that. All the doctors I know in my circle get vaccinated, right? So it doesn't make any sense to me.
I wonder, broadly, we're told that people who aren't getting vaccinated, a lot of them just don't have all the information. And, I'm not trying to cut them slack, but, is that the reason for these people? I mean, they're so busy, they're seeing patients, they're doing this, that, and the other thing that they haven't really done the deep dive into the information that they need to do. I don't know either why physicians or other healthcare professionals would not get vaccinated. Boggles the mind.
It would seem that there are mechanisms for them to get information, right? When new treatment modalities are approved, they're in certain places that they can look to. Now, Daniel always says that the information transfer isn't 100% efficient. So many doctors in the U.S., are still prescribing hydroxychloroquine, right? Because they just are not in the loop. So it's not easy. Maybe that's a part of it, Kathy. Yeah, I would not think otherwise. Yeah.
Yeah, I agree.
It's frustrating.
It's frustrating. It's also, one way I've been thinking about recently is just how scary pandemics are. I mean, we're talking today about how this virus can crush through the globe in a month or two. That's pretty disorienting. And so as humans, regardless of what we do for a profession, what is the first thing that we might draw on in a scary situation? It's not always, I think it's maybe not always the company line. There have been reasons in the last generation or two, or even longer, why you shouldn't trust that voice of authority. And so maybe especially in these impactful moments, falling on your family or your church or your sort of that could be a different source of information, which is a reason for that disconnect. here, but it's rough. And I was just talking to a friend who's a physician in Minnesota last night, and another friend who's an educator, also in Minnesota, and the twin crises that I think are emerging across the country and broader, which is what are we doing about supporting our teachers? What are we doing about supporting medical professionals who are now 2 years into this and wondering, why should I show up tomorrow? It's hard to open a school tomorrow morning. And that's whether Omicron goes away or the next variant, that's going to persist for years to come. And same in the medical profession. And that's another thing that sometimes I think when we get caught in these discussions or sort of the heat of the moment of who's getting vaccinated or who's not, we have some bigger issues here when it comes to that. Hopefully becomes, I think, a deeper part of the conversation if we're going to support folks who are kind of on the front lines in a sense.
Yeah, we're learning as we move in this now, because it hasn't happened since 1918 to this severity, right?
Yeah, agree.
Just one little P.S. Racaniello says that she's vaccinated and boosted. And my wife ran across a little thing today that points out that in England, booster is spelled BORCESTERSHIRE.
That's funny. Very good.
That's great.
I like that. Kathy, can you take the next one?
Yes. Sean writes, hello, Twivsters. I've only started listening recently, so apologies if this has been asked before. Is there a difference between, for example, fourfold and 4 times? I hear things like there was a 4-fold increase in something. I have always thought a 4-fold increase meant 2 to the 4th, so 16 times. I understood fold to be an analogy to folding a piece of paper over and over again, and the exponential increase in the resulting number of layers of paper. These terms are used interchangeably on the podcast. I started listening during the pandemic, and I think you all provide an incredible service. I have started Vincent's virology course on YouTube, and I'm fascinated. My education in biology ended mid-high school, so I spend a lot of time with the lecture paused and Googling for background. Love the show. Cheers, Sean. Well, to that second part, I say, hear, hear. If you stop and pause to
it's great.
Get the background, you're going to get that much more out of it. Is there a difference between fourfold and 4 times? I think of fourfold and 4 times as being the same.
Yeah.
And this idea that you have that when you fold a piece of paper, that's giving you an exponential increase is not right. It's just a fluke of the language that we say fourfold and it means 4 times.
But it's a beautiful interpretation. I love it.
Yes.
It's really great. It's just not how we use it.
Right.
And as long as we're on the topic, I would say that we throw around all the time 2 logs, 3 logs, 4 logs, that kind of stuff. Yeah. And a lot of listeners don't understand what we're talking about probably. A log is 10-fold. So a log is 10-fold, 2 logs is 100-fold, 3 logs is 1,000-fold.
In fact, I know of somebody who was in some kind of study section environment, but it was a very broad science-based thing, and they were with physicists, and the biologists were talking about a 2-log change, and the physicists were lost. Go figure. They didn't get it. So,
Nels, can you take the next one?
Oh man, you come on as a guest, but you're put to work here.
You're not a guest.
Absolutely. Adam writes, Dear TWiV team, such a privilege to listen to number 858 with John Mascola and to get a glimpse into the selfless world of the public health that protects us all. The discussion touched on evolutionary pressures leading to phase 1 candidates, biologic and immunologic surveillance, and the need to build sustainable structures that can be called into use. Around minute 5450, Rich raised the subject of surge capacity. And here Adam is paraphrasing. You want to be ready with science, tech, and manufacturing, but you can't build this capacity and just let it sit there for 20 years. You have to be able to shift the capacity to accommodate the surge that comes with a pandemic. It seems like a huge problem. And then John Mascola, your guest, responded, it's feasible if people are thoughtful and intelligent. One can build critical research capacity in various regions of the world where they can be doing immunologic surveillance. I guess Alan jumped in to say models from other spheres of activity suggest a way to do this. For example, volunteer fire departments, where volunteers are trained and then get called up as needed. John then referred to regional capacity. It should be possible to have manufacturing based regionally, which led to Brianne's comment about developing a platform which can then pivot to deal with whatever pandemic comes along. So Adam says, it struck me that this description of the public health system implies an organization with counterparts in the intracorporeal immune system, humoral and surface antibodies, memory cells and germinal centers, and just the like pre-infection modulation of immunity. I wonder if the presence of a pattern might offer a little information. Perhaps public health officials developing a structure for research have been following the same evolutionary pressures that have brought about mechanisms for immunity. This is not a testable scientific argument, of course, but there may be clues of use in pandemic planning. All the best from London, where, as this correspondent said last week, we expect the weather you had on the East Coast 2 weeks ago. I'm guessing in line with episode 855. And that's from Adam. Yeah, I have to say, I really like this analogy. Whenever I'm out driving on a highway and you see, an abandoned car on the shoulder of the road, oftentimes it'll have a marker on it where, someone as part of the Department of Transportation, right, has come by and flagged that to say, okay, we've got a breakdown on the road. And then they don't actually take the car away. But now that it's marked, another tow truck comes a few days later and says, oh, that mark means I'm supposed to tow this car away. And this to me feels like adaptive immunity, where we have antibodies that bind to cells that are containing foreign agent. And then we have I think there's some really interesting echoes at different levels of biological organization, all the way from the molecular, from immune systems up to human behaviors. And so it's fun to think about. But with any time, I think when you're drawing analogies, they can break down as well. And so I think you want to take a little grain of salt there, but it's a fun way of thinking about things for sure.
All right, last one from Ken, 54F12C and sunny in Glendale, Arizona. I'm fairly new to the YouTube channel, but the series of podcasts has quickly become one of my favorites. I apologize if the following feature exists. I have not been able to find it either on YouTube or microbe.tv. The premise of most of these shows involves discussing and dissecting various papers. As a listener, it's interesting to hear subject matter experts confer on various aspects of the paper. I want to be more than a passive listener with these podcasts. I was wondering if there could be an email group established that would connect with the audience and assign the papers that will be discussed before the show. I understand sending the actual PDF may be problematic, but at least the information sent and those with access to databases could acquire the paper individually. I would use that opportunity to look at the paper, write notes in the margins, as we all do, and compare my notes with those of the experts. I'm sure there are many other benefits to this approach, but this may be another step in your vision for the program to educate people on virology and science, and indirectly reinforces the approach by reading journal articles. I'll stop the message there because I want to respect your time. Thanks for all you do. You and your colleagues are having a positive effect on the world, Ken. So, Ken, I have for some time now wanted to start a Discord channel, which is a place, focused on a topic where everyone can go. We could post the papers there and people could then have them as you see. And then they could also have a discussion. So Discord is meant to have a discourse, right? And so I did make a Microbe TV channel, and I never pushed it further than that. But I would like to do that, and people could go independent of us. We don't have to be there. They could have discussions, and they would have the papers to do as you say. So I will try and get on that, Ken. I'll put it on my list. I need to make lists, but I haven't.
In the meantime, I don't know if this is possible with the way you list the shows on YouTube, but the way they show up on the website, all of the show notes are there that include the papers.
That's correct.
So as hard as it might be to do, you could, before listening to the episode, go to the show notes, find the papers that are going to be discussed, and read them, and then go to the episode. I realize that, that's like withholding the candy that's sitting there right in front of you, okay, to do your homework first. But
That's a good idea. I hadn't
One way
thought of
to
that.
approach it.
I hadn't thought of that.
So I actually do something like that with some assignments where I assign these papers, and that's where I get them. If it's on an episode I wasn't on, is I just go to the show notes and get the paper, and then assign the episode to my students.
And I think in the show notes, the papers that are going to be discussed are pretty clearly highlighted. There's a lot of links there, but you can I think identify the papers that are going to show up.
And then, of course, Jolene puts the timestamps on, and you can go exactly to that point in the episode if you want. Yeah, that's a good idea. So both of those things could work out. All right, let's do some picks. Nels, what do you have for us?
Yeah, so my pick is a book and a book review, actually, that we covered as well on the upcoming about to release episode of TWiV, episode number 74. And so this is Racism, Not Race by Joe Graves and Alan Goodman. The link that I'm including for my pick of the week is from Agustin Fuentes, who did a review of the book in Science. And this is, Vincent and I had a really, I think, positive and important interview with Joe in our episode of TWiV, thinking about the differences between social constructions of race and blowing away the notion that there's a biological basis to this. And this is, I think, an important time for these conversations, both in the fields that we're in science, and in particular in my field, in evolutionary genetics, evolutionary science, human genetics, but also just kind of across the board. And so Joe told us a little bit about his journey through academics and in evolutionary biology in particular, has some important perspectives, and is, I think, casting some light on some of the imbalances when we look at representation in evolutionary biology and all of science, really. And so this book, it just came out kind of at the turn of the year, is really interesting. The audience is kind of everyone who's interested in, stepping through racism, cutting through this, and the idea of race science as it's reemerging in different forms. And so maybe start with the review here at the link, and then big recommend for me. I've really enjoyed the book. It sort of weaves together the history of racism, of race science. It takes the scientific, also sort of the cultural, and then Joe, who's on faculty at North Carolina A&T, talks about his career, kind of brings in his experiences, and then Alan Goodman, who's at Hampshire College in Amherst, Massachusetts, an anthropologist. They sort of weave their stories together in a really compelling way. And so big recommend. And if this sounds interesting, maybe take a look at TWiV 74 as well, to hear from Joe himself on these topics.
Thanks, Nels. Rhian, what do you have for us?
So this is an article that I read and that I sort of got a kick out of. It's called Groundhogs Emerge from the Scientific Shadows in the New York Times website. And I just thought it was funny, of course, first of all, because it's today on February 1st, getting ready for tomorrow, Groundhog Day. But this talks about the animal behavior of groundhogs that had not previously been studied, and new insights that have been studied, and particular into how social these animals are, how much we did not realize that groundhogs were very social, and how much of that has been emerging. And the entire time I read this, I was thinking to myself, wow, there's got to be a lot of spread of viruses among groundhogs if they're living in these large colonies. The entire time I read it, I was starting to wonder about what it would mean about spread of viruses among the groundhog colonies. So I highly recommend learning about groundhog animal behavior, and then we can wonder about viruses together.
Yeah. The one holiday for an animal. How about that, huh? That's pretty cool. Rich, what do you have? I'm sorry, Kathy, what do you have for us?
I picked something that's from Smarter Every Day podcast, episode 268. And I think we've picked Smarter Every Day before, but I don't know how YouTube just feeds you random things. So this is about a zigzag Coast Guard search pattern and a Victor Sierra search. And if you think about it, if you drop something on land, you just retrace your steps to go find it. But in water, with currents and things going on, it's not so simple. And so there's this thing called a vector Sierra search, which looks all the world like the radioactive symbol if you just draw it straight on paper. But then when it's executed on the water, it doesn't look quite like that. Anyway, it's pretty cool. And he does a really good, excited visit with all these different people in the Coast Guard. He calls them coasties. And partway through, they teach him how to whistle. And it's something you can only do one time. They show him how to do it. And you can see whether if you do it that first time, whether it works for you. But it's a pretty fun podcast. So check it out.
Cool. That's very neat. Rich, what do you have for us?
Well, as I said, Kathy has gotten me re-addicted to Astronomy Picture of the Day. So I picked today's, which caught my attention, that's called Moon Phases 2022. And this is it has a timeline. It has a big picture of the moon in the front and shows you what the phases are going to look like at different times of each month this year, with a timeline going on in the back, and another little thing about the moon tilting and all of this kind of stuff. And I thought it was just really nicely done. But it also, if you read the fine print, the pictures were taken by a thing called the Lunar Reconnaissance Orbiter, which I didn't know existed. And this is a spacecraft that was launched in 2009. And as is sort of typical for NASA, it had a mission that was supposed to last like 2 or 3 years, and it's still taking pictures. Okay. And this is a satellite that orbits the moon and collects data, pictures, and all kinds of other stuff, among other things, so that they can be have the lay of the land should they decide they want to visit again. So it's kind of a pick within a pick. The moon phases are cool. The Lunar Reconnaissance Orbiter is cool.
That's very neat.
APOD's been good lately.
Oh, APOD's been great. Great stuff. Did you see that sun thing yesterday or the day before?
Yes.
Holy cow.
Good.
Has everyone been following the Webb telescope?
Yeah.
Oh, yeah.
Fingers crossed that they can get that thing up and running. So far, so good, it looks like.
So far, so good.
Amazing. All right, my pick is the Fourier transformation. And you're like, what? Who cares?
Oh, man. This takes me back.
So yesterday in my virology course, first time in person since spring 2020. It's been 2 years. But it's funny. I had to go to the undergraduate campus, and now they have all the locks on the doors. Every building used to be wide open. Anybody could walk in. Now all the buildings are locked. And I'm pulling. So I walked around the building. And then I saw there's a little card thingy there. Now you put your ID on it. So I put my ID on it. It didn't work a couple of times. And I waited there till a student came in, and I walked behind them. And then I get an email from the university. You attempted to enter a university building without your verification. You have to do a daily symptom verification on your phone, and that links to your ID. And I totally forgot because it's a different campus. I never go to it. I haven't been to it since January of 2020. Anyway. So yesterday we talked about structure, and part of it is cryo-electron microscopy to solve virus structures. And the way you do that is you take pictures of virus particles that have been frozen in aqueous solution to have a little contrast. You take many, many pictures, which are all in different orientations. And then it's like a CAT scan. You combine all of those to form a three-dimensional image. And the way you do it is by calculating the Fourier transform of each image, and then figuring out the centers, aligning them all, and doing a reverse Fourier to get the virus particle. All right. So a Fourier transform is basically an equation that can transform one kind of information to another. Officially, it will change space and time information into spatial or temporal frequency, and it can be applied to anything. Can be applied to music. For example, if you go to the Wikipedia page, the expression of a musical chord in terms of the volumes and frequencies of each note, right? That would be a Fourier transform. So how to make this accessible? So I have two places, and one I've had for many years, over 20 years, this website, Kevin Cowtan's Book of Fourier. So Kevin is at the University of York in the UK, and he's a structural biologist. It's called a book of pictorial 2D Fourier transforms. These are just two-dimensional. When we do viruses, we do three-dimensional. But for example, there's a number of different pages. One of them is called the Fourier duck. So he has a picture of a duck and it's Fourier transformation. And of course, they don't look anything like each other, right? And he has a series of examples where he has a cat also. He has a Fourier cat where a tale of two cats. So they have a cat without a tail, and he tells you how to manipulate the parameters of the Fourier to get the tail back, right? So he uses this to explain how it works in crystallography. So it's really a lot of fun. He has also animal liberation page, which is more ducks, and the gallery. He also has pictures of dots and how they would be a Fourier transform, so you can get an idea. So then I found a YouTube video which is live Fourier transform. This guy must be a teacher somewhere, but he takes his video and he's somehow putting it through a Fourier transform, which he puts next to him, right? So he said, okay, this is me, and that's my Fourier transform, which of course doesn't look anything like him. And then he takes a series of geometric objects, like he'll take a black dot on a white background, and it gives you a pattern of concentric circles. And then he'll do a white dot on a black background, and you get the same exact pattern. And he said, you can't distinguish, and that is the phase problem. And that's exactly what you solve in solving virus structures, how to align the phases, right? Anyway, I don't really understand the underpinning mathematics, but these two sources will kind of get you on the right track to figuring out, how we use this. I mean, this revolutionized. Someone called it the most important function in mathematics, right? This ability to do this. It's applied to so many things, and it revolutionized cryo-EM. You couldn't do it without this, right? But it was not made for cryo-EM. It was developed by Joseph Fourier to study heat transfer. Okay, and Fourier was living he lived from 1768 to 1830, and his function is used to solve virus structures. Isn't that amazing? I just think this is so cool. All right, Fourier transformation. Then we have a listener pick from Dusty. Hey, TWiV team, thanks for all your hard work with science communication. While I know I will never have time to listen to all of the back episodes, I'm glad to know that you have all probably covered any aspect in virology I may be interested in if I ever want to learn more about a topic. It has been so helpful to hear other scientists work through an area I have very little experience in, but a newfound interest over the last 2 years. Now, by the way, Dusty, there are people who say they've listened to every episode. While I was listening to episode 860, you all were talking about science funding and the differences between academia and the NIH and others. And I was reminded of another podcast I had heard recently discussing about how NASA is working to address the biases in which projects are funded or given telescope time. With the exciting launch of the James Webb Telescope, hopefully some other disciplines will be inspired to improve the ways they fund projects. So it's a problem in not just biomedical science, obviously. Fighting bias, this is an NPR article, Fighting Bias in Space. When there's a new telescope, who gets to use it? Kind of reminds me of the electron beams, right? When you want to solve your structures, there's accelerators and you have to get time on it. And how do you decide how to do that? It's really interesting. So this is a podcast. So
excellent.
I like that very much. Thank you, Dusty. That'll do it for TWiV number 861. You can find the show notes at microbe.tv/TWiV, or you could go get the papers, as Rich said ahead of time, read them, and then listen, or you're probably not going to do that. If you want to send us a question, comment, or pick, TWiV at microbe.tv. If you'd like to support us, we have a page listing the different ways you could do that, microbe.tv/contribute. And of course, now we are a 501c3, so your contributions are tax deductible. Our guest today, Nels from Selfcalibration.org in the University of Utah. Great to have you, Nels.
Yeah, thanks so much, Vincent. It's just really fun to be together and kick some science around. Can't wait to do it again.
I liked having crossover podcasts. We have to do it more often. It's a lot of fun.
Count me in anytime.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Condit's an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. Great to see you, Nels.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. Thanks for being here, Nels. That was really fun.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.